Hello everyone, good morning, good afternoon, and welcome to today's webinar. My name is Carmen Guta and I'm your host from Technobis Crystallization Systems today. Our webinar on controlling polymorph impurities in pharmaceutical processes is being presented by Jude Eames from a team leader solid state from Eurofins CDMO Alphora. Today's webinar was possible with the help only from Eurofins. Eurofins, a great partner to us for many years now. They have kindly agreed together with June today to bring these case studies to you in this webinar. But before we start, I would like you to tell you a few technical things about the webinar. Is being recorded and will be made available in a few days on our website. Also, you will receive an email with a link where you could review this recording this webinar. If you have questions, please make use of the chat function, Q&A function of this platform. I will read all the questions at the end of this webinar to Jude and try to address all of them today. If afterwards you still have questions, you can contact us directly at info@crystallizationsystems.com or Jude or Eurofins via their website. And I think Jude will also share with you some contact details. You will hear also a bit more about Eurofins and how you can get in contact with them if you need to find out more information. Before we start, to introduce who is Technobis Crystallization. Technobis, we are a company, a Dutch company in the north of the Netherlands, world-leading technology provider for solid-state research, process development, and formulation. We are on the market since 2005, with by now 2 portfolios, one on crystallization screening. Our webinar today and what Jude will present is more related to the crystallization portfolio, but also since 2 years ago we have a reaction screening portfolio with the Reactol product. The Reactol, for those that did not hear about this product, is a multiple reactor system, as you can also see here, 5 reactors in parallel. And the beauty of it is that it has automated sampling. So while you perform your reaction, can also sample. You can program it, and not only sample, but the sample is immediately quenched and diluted and transported to the LCMS HPLC vials that later on you can put in and for analysis. Is an instrument that addresses any type of issues or case studies for route scouting, reaction screening, reaction optimization, reaction kinetics. I have personally experienced quite a lot of reaction kinetics in the lab at our customers. And focusing on today's topic and the crystallization portfolio, some of you might already know the Crystal16 or the Crystalline, even if I see the names that joined us today. If after this webinar you think that you'd like to test one of our instruments, as I mentioned, you can contact us via the website or at info@crystallizationsystems.com. Please remember the Crystal16 is your instrument for solubility determination, temperature dependent and solubility determination, metastable zone width, and any type of solid state screening. The Crystalline comes with the camera technology, or if you have a Raman, you can also Raman probe, you can connect it to the Crystalline, and you can with it practically develop, optimize your crystallization formulation processes. And the crystal breeder works at really small scale, microliter scale, and is really the gadget instrument that I like to call it, that addresses single crystal growth. And with that, I don't want to go too much into detail about Technobis, and I want to give the floor to our presenter today, Jude Eames, team leader solid state at Eurofins CDMO Alphora. She has had the privilege of collaborating with a diverse range of clients and projects at Eurofins, has over a decade of experience in characterizing materials and more than 5 years in pharmaceutical research. The focus is on ensuring robust processes that consistently deliver high-quality materials, overcoming unique challenges with innovative solutions. Outside of work, Jude enjoys spending time with a cat and playing Aussie rules football. And I think this is Jude. You were very shy introducing yourself in just a few words. We know each other for quite some time now, and I know that your experience goes much further than these few words. And I truly appreciate that you took the time to make this presentation and to present today and to bring these case studies to our attendees today. I will stop sharing and I give you the floor is all yours, Jude.
Perfect. Let me just bring up my screen. I am actually here with my cat today, so she might make an appearance. Okay. Perfect. All right, let me put that in presenter mode. That's up and good?
Yes, perfect. Thank you.
Thank you, Carmen, so much for your very kind introduction. Yeah, it's been a great relationship so far, and hopefully continues to be. I'm sure it will. So I'm Jude Eames. Just to give you a little bit of context about who I am, where I come from, and what we do. So our company is Eurofins CDMO Alphora Inc. So we were originally founded in Canada as Alphora back in 2003, and then we were acquired by Eurofins in 2017. That acquisition allowed us to invest in more capital, and while we were previously an excellent research and development in pharmaceutical manufacturing, it allowed us to introduce the solid state labs, which is where I came in, and we were able to do a lot more with crystallization and testing where we previously had to outsource. We have many services. We are a large company at Eurofins CDMO Alphora with drug substance, solid state R&D, biologics, and drug product. You know, we can do a lot with what we have, but I'm mainly from solid state R&D, and we'll mostly be talking about drug substance in this presentation today. When I'm talking about solid state research and development, there's many things you can do with that in pharmaceutical development. So a couple of things we do are high throughput screening, characterizations of materials with all the different instruments we have, and a little bit of crystallization engineering. So I'll be talking about crystallization engineering specifically because it relates to the crystallization tools that Technobis provides, and hopefully I can give you a little bit of information how we use it and how you might choose to use it in your company. To do that, I'm going to go through a case study that has some really good examples of how we've used these instruments and how they've really helped us understand what's going on. So in this case study, our client came to us and they had an active pharmaceutical ingredient, or an API, that's a white powder, and for their drug delivery, they had to melt that powder into a nice translucent melt. They found with the batches that we gave them early on, there was an issue with some batches having these unmelted flakes. So you can see in this image with the scale bar, there's a transparent melt, but it has these white unmelted particles in it. And that was not suitable for their use. It was entirely undesirable. So we need to go back to the drawing board and figure out how to fix this problem. We had some goals as we were doing this development. We wanted to solve that melting issue that was caused by this polymorphic impurity, which I'll get into some more. We also had to make sure that we maintain the product quality. So that means the chemical purity. We weren't willing to sacrifice chemical purity in order to reach our other goals. And always, because we are working with our clients, they're the ones who set the budget and the time frame, so we had to be conscious of that and work within their expectations. So I'll give you a little introduction about how we make this powder. A lot of it is confidential information, so unfortunately I will be vague on certain things, but I'll give you enough information to understand the key learnings. So we're going to talk about the final isolation of this material, which is a crystallization by cooling. So what that means is you start off with your kind of your reaction mixture, or your soup of all your you might have excess reagents, impurities, and the thing that you actually want to isolate. So you heat that all up and do a filtration. This is very important in pharmaceutical processes to do this at the last possible step, because it gets rid of any solid impurities that might have been introduced by reagents or equipment earlier in the process. And obviously we want nice, clean material if it's going to be pharmaceuticals. We then cool it down, and as you cool down this solution, you ideally want your product to crystallize out into solids in solution, and then you hold it at that cold temperature and then filter off the solids, and there you go. You've got your nice white API powder that you can give to your client and say, here, we managed to produce 10 kilos of it. Before I go more into the specifics of this case study, just an introduction into polymorphism. So you have your API, your active pharmaceutical ingredient. And when we're talking about polymorphs, we're talking about different structural arrangements of that same drug molecule. So up the top line there, you can see you have amorphous, form 1, form 2. It's all the same pure chemical, same molecule. It's just in different arrangements. So in amorphous, you don't have any regular structure. And the difference between form 1 and form 2 is that they're arranged in different structures. You can also create different crystal structures by incorporating things like counterions, solvents, and coformers, as you see in the images there. And why we care about polymorphs is that it can change the physical properties. In this case, we're really interested in that melting point. But more generally, when we're talking about polymorphic control, it's important because it might change things like solubility or bioavailability. And when you're talking about a pharmaceutical, that can have an impact on the patient or the recipient of that pharmaceutical. So we definitely need to make sure we have 100% control of that, because we want to make sure that everyone taking any medications is completely safe and getting the exact dosage that we want them to get. So in our particular process, we had done polymorph screening in the past, and there's over 20 polymorphs that are well known. But in this particular process, there's only really 4 that we care about. So we have our target polymorphs, which are these 3 here, A, B, and C. They're in green, they're good. So from the cooling crystallization, we initially get out this form A, which is a solvate. It's not very stable, and as we dry it, it goes through form B, which is metastable, and then eventually ends up as form C, which is our stable final dry product. That's what we want to end up with, pure form C, in our final white powder that we isolate. Unfortunately, there is this polymorphic impurity, form D. It's popping up in our final product, but we don't know if it's coming from early stages or late stages. That's something that we had to figure out during our development in order to get rid of it. We do know some things about these different polymorphs. We know a lot of things, but the relevant things to know are they have very different melting points. And that's what makes this form, our target forms, the target forms, because they melt really easily for our client. Our client was melting them at about 170 degrees. Form D actually is more thermodynamically stable, so it has a much higher melting point. This generally means that it's going to be the most stable and easiest to isolate polymorph. So if I could, I would suggest to our client, hey, form D is going to be much easier for your process. Let's just go with that. But unfortunately, for the uses that they want to use this material for, it's much too close to that decomposition temperature. It's so stable that it doesn't want to melt. And it's eventually, if you do get it to melt, you might be degrading your product with all that heat. So we do really need to target these lower melting point polymorphs that we have here. The biggest issue we ran into in this particular case study is that we weren't able to detect this impurity. So usually what we would do is PXRD, or powder X-ray diffraction. That's a technique using X-rays to detect the crystal structure. And usually you'll see, if you do have an impurity, you'll see peaks poking out, and you can see if you've got a mixture or pure polymorph. When we were looking at this unacceptable melt material, we weren't able to detect any of that impurity. We thought maybe it was the power of our X-ray. We thought maybe we could get better penetration if we sent it to a synchrotron X-ray facility. So we sent them a couple of good, bad reference samples. And again, they weren't able to detect any difference. We did also try a couple of other techniques, including DSC, or differential scanning calorimetry, but it was kind of hit and miss whether it was able to detect it, mainly because sample sizes are quite small for that. And it seems that this is not a homogeneous mixture throughout. They're quite large flakes, so it was very difficult to determine from our testing the actual properties of the bulk. So we came up with our own bulk test in order to detect this Form D impurity, and we called it the melt test. So what we did is we put our bulk powder between two glass slides and put it on a hot stage. So that allows us to heat up our material to 170, and we're able to look in a microscope, specifically a polarized light microscope, and it makes any unmelted crystalline material stick out. As you can see in the picture, you have those bright white spots which indicate unmelted material, which we know is our Form D polymorphic impurity. So that's great. We came up with a test to kind of uncover this hidden impurity. So now we had to use this test in our process to try and figure out how we could remove the impurity from the final. As always, when you're talking about a cooling crystallization, step one for polymorph control is always seeding. So when you're seeding, it's like the name suggests, you're kind of planting seeds in your process, or adding solids to your solution, and it works as a template. So it kind of tells the solids that are forming out of solution what crystal structure they should have. So when we go from, in this image, point A to point B, you'll see that you travel as you cool down, so move to the left. You go through the dissolution line to this metastable zone. So the metastable zone is where you want to be seeding. If you seed too close to point A or the dissolution line, when you add your seeds, they're just going to dissolve and it will be like you never added them. So there was no point in doing it and you won't get that templating effect. If you seed too close to point B or the nucleation line, you'll find that solids have already started coming out of solution, and so whatever comes out of solution might act as the template instead. So for our process in particular, we wanted to characterize that dissolution, nucleation line, and the metastable zone so that we could get better control of the polymorphic form. So we did that on the Crystal16. It's our go-to instrument whenever you want to measure solubility and determine the metastable zone. But an important factor here is that you should be using materials that are typical of the system that you want to understand. So early on in development of this process, we had used isolated solids in neat solvent. So that's our kind of clean system. That's really great when you have a lot of isolated solids. You can easily measure them out accurately, and they're very easy to work with. I definitely recommend doing this if you're going to be trying a lot of different systems. It's early development. You want to try 10, 20 different solvents and look at the metastable zone width of each solvent to selecting your solvent. That's absolutely the way to go. But when you're in later stage development and you want to have more control over a particular system, like we do in our case, we've already locked down our solvents and our process. That's when you might use this crude slurry mixture method. It is more difficult to handle because you're dealing with kind of your reaction mixture, which might be solids and solution. It's hard to homogeneously and representatively subsample them, but it will give you a better idea of how your process actually responds to temperature with all those impurities present. Those impurities can have a massive effect on that metastable zone width. So this is our example data that we collected. Not the best fitting of the lines, but again, that's because it's this slurry mixture that's very hard. This is what real world data actually looks like. And we were able to determine that the dissolution line, nucleation line, and ideal seeding line. For our process in particular, we're interested in that one concentration. That's what we want to understand at that 20 volumes mark. And so we can see there's a kind of a plus minus of 1 volume there because we don't have 100% control over our concentration. So we want to give ourselves a buffer zone. So with this data, we were able to say this is the best seeding zone, about 44 to 47 degrees Celsius. And that means that we'll be right in that metastable zone, and our seeds will be able to act as templates and we'll have better control over the polymorphic form. When you do the same thing with the kind of clean system, you find that the ideal seeding temperature was actually a lot lower. And that's what we found with our original data is that when we were seeding our actual process, solids were coming out of solution before we were adding our seeds, which is not going to work. So I just wanted to point out that you definitely want to make sure that your system is representative of your actual process. So we implemented this seeding point. It did help with the polymorphic impurity, but it didn't 100% solve our issues. We were still seeing Form D in the end of our final product, so we had to keep working on that development. We made some observations when we were doing these melt tests of a couple different batches and testing our seeding. There was a sample that was submitted and tested one day, time point 0 initially, and we couldn't detect any of this Form D impurity in it. But when it was tested again the next day after being left at ambient conditions in the lab, it had a significant amount of Form D. So that kind of raised a question about the polymorphic stability and changes that might be happening. And it was also interesting to note that we suspected it was because there was still some solvent from our process present in those solids, because we hadn't seen that before. It was just this particular one. So we decided to do some investigation using the Crystalline from Technobis. And actually, we were doing this experiment on the demonstration model that they lent to us. And it led us to understanding that it could be very useful to us in our lab. And I'll show you some of that data that we collected. So this profile, we were in control of the reactor temperature. So you can see in the red and orange lines, we start off at 20 degrees C, cool it down to -5. Into the reactor, what we had was our Form D solids, so our impurity that we don't want, and we just had them in our reaction solvent neat. And the great thing about the Crystalline is that you're able to get images. So throughout the entire process, it's saving images and you can go back and look at them. So we didn't see any changes initially on cooling from 20 to -5. But what we did see, interestingly, is that after about 60 minutes in the slurring at -5, we did start to see a small a small change in the morphology. So from points A to D, you see that they're kind of a regular, chunky, round morphology. And then right here at point E, we start to see our first inklings of this needle-shaped morphology. And then as we go through to 75 and 90 minutes, we start to see that needle morphology really dominates. And so what that's showing us there is that we're getting a polymorphic conversion from Form D, our impurity, to Form A, our solvate that we're getting in solution, which is great. That's a light at the end of the tunnel. We can convert what we don't want into what we do want. Whenever we see this, so with the observations that we saw in the lab and in the Crystalline, it lets us know that we have an aneotropic system. So a monotropic system, at all temperatures, you're always going to isolate a single form. It's more stable. But in aneotropic systems, there's some crossing point temperature that we call T0, at which the lower energy polymorph switches. And so we wanted to find out what that T0 was to help us isolate our form A and fully understand what was going on there. We did this through just a couple of experiments at the 1 gram scale. We did it at the same concentration as our process, so it would be more directly related. And we didn't need to do the melt test because we don't need to know the trace polymorphic forms. We want to know what the bulk is doing, you know, that tipping point at which we see a great amount of change. So we did a couple of experiments at a handful of different temperatures, 20, 10, 5, and -5. And as you can see, they had different results. So form A, we saw at benchtop convert to form D. We didn't see any conversion at 10 degrees, even though we let it stir out for 1 week. At 5 degrees, we did start to see conversion, but it was slow. It was taking a few days just to convert that 1 gram of material. And then when we go to -5 degrees, we're seeing conversion within a few hours, which is similar to what we saw in the crystalline. So now we understand where that T0 or that crossing point is, but now we don't actually care about that because it's more important the kinetics. So the difference in the Gibbs free energy is driving the rate at which we get conversion. So now we know that we want to go as cold as possible, and that will help us with our conversion to our ideal polymorphic form. So now that we understood that, we have to implement it in the process, but we did find it had an impact on the purity. So this is our original, heating and cooling down to 20 degrees C before isolating. If we implemented that -5 isolation temperature, we actually found that it had a massive impact on the purity, lowering the impurity. And that's because we have those impurities present in solution. And if we cool down too much, we end up isolating our impurities along with our pharmaceutical that we want to get. And the way that we overcame this to get both polymorphic and chemical purity is we did a 2-stage process. So we isolated first our material at 5 degrees C, which gave us good chemical purity, not the best polymorphic purity. And then we put it back into a neat solvent. So those chemical impurities had been removed by a filtration. And we were able to reslurry them at -5 and get full conversion to our desired polymorph. So as an overview of the case study, we had an original process that gave us a material that was not suitable for our clients, and we weren't able to detect why that was. And in our development, we were able to detect the issue, work on a couple of experiments at a small scale, and be able to develop a process that worked at scale to give us chemical and polymorphic purity. So there's kind of 3 key takeaways that I wanted to demonstrate with the case study. Number 1, just because you can't see it doesn't mean it's there. I would always recommend using multiple test methods. So don't just rely on PEXRD, maybe do DSC, SEM as well, because you don't know what's there until you see it. And you might even have to come up with your own test methods like we did with the melt test to fully understand what you do have there. Number 2 is always try and use process representative materials. It's fine if you don't have your process locked down at the start, but you might want to retest if you have changes in your process, because it might have impacts on things like your solubility, that metastable zone width. So consider what has changed in your process when you're trying to collect data. And number 3, it is possible to isolate the less thermodynamically stable polymorph. Not that we recommend it, but if you have to, it is something you can do. And if this is something you want to do, we can help you with that crystallization engineering. So thank you, and I'm sure you've all put some questions in the chat that Carmen will go through in just a minute. But if you do want to contact us, you're welcome to. There's a QR code here to our website. You can visit us at eurofins.com/CDMO, or reach out to me at jude.eames@bpt.eurofins.ca.com.
Thank you very much, Jude. You already have some applauses coming up. I don't know if you can see that. Thank you very much for sharing this case. Really interesting. And to be sincere, it's It's nice to see. So one form that looks to be the more thermodynamically stable form, and usually you want to go for that as a company and so, but actually giving so many problems. I see questions are coming up. I will start to read them to you. Somebody is asking, it has a question about slide 16. You have there, I think, also the metastable zone. And the question is related to, is the solubility data shown here for form D or form C?
This is for form C with a caveat that it might have impurities of form D. This is representative of our process, which is mostly form C.
Yes. How different was the metastable zone width for form D versus form C? So,
yeah, unfortunately, form B and form C, whenever we put them in solution, they actually, after some time, convert back to the solvate form A. So in the process, we have form A and we're isolating form C. But if we try and collect solubility data, it's likely that it will, after some time, convert back to form A, which is fine because that's what we have in our process anyway. We're isolating it as form A first and then drying. So we can't actually observe much about form C and form B in solution, and there's not much that we can observe about form A being isolated. Unfortunately, it's a bit difficult to collect data on these unstable polymorphs.
Yeah. But form A, you are saying that is a solvate, and it's somebody is asking what is form A. I see it here on your slide that is a solvate.
Yes.
But what type of solvate? Can you bring that on a in a process? Make it.
So
it's not medicine, or is it? I didn't.
It's a specific solvate, but I don't want to go too much into the specific process. Yeah.
Okay.
But yeah, it's just your API molecules incorporating that process solvent into the crystal structure.
Okay. Somebody has a few questions related to the enantiotropism. Yes, if you can give a bit more information there. Yes. They are also saying that if we really want to talk about enantiotropism, both forms should be anhydrous. So not only form D, also form A.
Yes. So if you do, generally when we talk about polymorphs, they talk about true polymorphs, which don't include things like solvates, co-crystals, and this kind of thing. And yes, generally when you're talking about enantiotropic systems, you are talking about true polymorphs where you're just talking about arrangement of IPAs. You don't bring the solvent into it. But it's something that is also observed in this case study. We do have a system where temperature changes the stable form that we're isolating.
More a general question. How can hydrate formation be suppressed during co-crystal screening when a stable hydrate phase preferentially crystallizes despite the use of dry conditions and anhydrous solvents? Any insight on isolation strategies?
I mean, yeah, we have something similar here where we do end up isolating a hydrate, and even with drying it stays a hydrate and moves through the solvate hydrate. If you do need water there in your process, it might be coming from the atmosphere or the air, so you can try running them under nitrogen, which is very difficult to do at the screening stages. I would say, again, it seems like a similar experience to here, where our form D or the hydrate is very stable and maybe not allowing you to see all of the possibilities. But yeah, you can try nitrogen or argon blanketing and using anhydrous solvents and molecular sieves to really pull all the water away from the system. But I mean, if you're going to that much trouble at the small scale, you're obviously going to run into issues at a larger scale, and maybe it's not the best forms to be isolating. Maybe you just need to let the hydrates exist if they want to.
Yeah. And then somebody is once again, but now you just put these slides. I think this slide, many people have questions related to the conversions between the a few forms of this API. So Andy has a question, says, do I understand correctly that the solvate form A is turning into form C during drying? But if you cool down solution sufficiently, the non-solvated form is forming?
No. So if you cool down in solution, you do get the solvated form. It's just that when we take it out of, it's kind of an interesting case here, in that in most solvents, we're forming form D. It's very stable. If, you know, out of the hundreds of screening experiments we did, most solvents are going to give you form D. And in fact, even if you use water or wet solvents, or if we add water into this process, we end up with form D, which is strange because we're aiming to get a hydrate. But we think what's going on, again, we're limited because everything's a little unstable, so it's difficult to collect data on each of the forms. But we think what's happening is the solvent is kind of templating in that crystal structure. Then as we take it out, it's being replaced by water during the drying process.
Yeah. Have you tested the critical water activity between form C and form D?
We have, and we just found that the more water there is, the more likely we're forming form D. It did increase trace amounts of this form D impurity. We don't know the exact critical water activity because it's difficult to measure the trace impurities of form D that are created by small amounts of water. So we just decided to completely avoid water as much as possible. We didn't need to dig down until that exact threshold.
Okay. To avoid form D. And somebody, maybe this is the last question that we will take, is asking about the crystalline instrument system. I think you have showed it. Now, what is your opinion about this instrument? Why would you use it? And what do you like from it?
Yeah, initially we were struggling to come up with ways in our projects that we could use it, but now that we've gotten used to it and kind of figured out its best attributes, we use it for most projects now. For us, it's a programmable small reactor. So while you might have programmable reactors that are 100 milliliters or 200 milliliters or 400 milliliters, we often don't have enough material to screen different solvents, different seeding temperatures, different solvent compositions. Whereas when we have this small programmable reactor, we're able to try more conditions with less material. So we're being more efficient with the material. And because it can run overnight and take images, you can see when things are oiling out or changing morphology or becoming stuck to the vessel walls. So we're able to increase our throughput and get more data from less material.
Do you make use of the cameras?
Yes. The cameras are very helpful for us. In this situation where we saw a change in morphology, that's quite rare. Typically what we're seeing is a change in the phase. So typically we have issues with oiling out or our solids being in a liquid phase. And so we really need to investigate at what temperatures and solvents we're getting solids and when we're getting liquids. And we can see that with the camera.
Okay. Thank you very much, Jude. I think this is the last question that we will take. Once again, thank you for bringing this case study. And as always, Eurofins goes always not one step more than a few steps further for their customers. Even you have mentioned synchrotron data. So definitely the quality of the work it's high at Eurofins. If anybody has different questions after this webinar, please feel free to contact Jude directly. And your email address was jude.eames at...
Yeah, I can drop it in the chat, I think.
Yes. Or eurofins.com/CDMO on the internet. You can contact them directly. Oh, yes, I will put it in the chat with the Q&A at bpt.eurofinsca.com. Here, I will put it once again in this one for everyone. So, but also, if anybody has any questions, we can also from Technoviz forward them or to make the contact between potential customers and you, Jude. As somebody says here, thanks a lot for the webinar, super informative, well presented, and addressed all the doubts. I think Vishal has really concluded really nicely. Thank you for that. Once again, thank you, Jude. And have all a good day and afternoon, evening in Europe.
Thank you so much.
Bye.